and glucose pathway. This is the phospholipid bilayer of a cell, such as a skeletal muscle cell, cardiac muscle cell, adipocyte, hepatocyte, or endothelial cell, certain neuron in the brain, or a pancreatic beta cell. Embedded within it exist various proteins, one of them being the two monomers of an insulin receptor protein. The monomers of the protein are made up of two subunits, the alpha subunit in yellow, which extends into the extracellular space, and the beta unit in pink, which extends through the cell membrane. Also embedded within the membrane is PIP2, a lipid found in membranes that plays a role in signaling. It can be broken down into smaller molecules that help it control processes like protein activation. Insulin is a hormone made by the pancreas that helps cells absorb glucose from the blood for energy or for storage, and can be found in the extracellular space. Insulin is produced when blood sugar levels rise, usually after eating. The pancreas senses the increase and releases insulin to help cells take in glucose. The free insulin molecules bind to the alpha subunit of the insulin receptor. This causes the receptor to dimerize, which means that the receptor pairs with another receptor to form a functional unit. This activates the receptor's tyrosine kinase domains, which phosphorylate or add phosphate groups to specific tyrosine residues on each other. This process is called cross-phosphorylation, which enhances the receptor's activity, allowing it to relay signals inside the cell. This causes downstream molecules to be recruited and activated, which means the phosphorylated receptor creates binding sites that attract specific signaling proteins. These proteins bind to the receptor and get activated, triggering further steps in the signaling pathway, amplifying and spreading the signal inside the cell. IRS binds to the active insulin receptor through its pH domain, which recognizes the specific phosphotyrosines on the receptor, anchoring it for phosphorylation and activation. IRS binds to P85 using its phosphorylated sites, which P85 recognizes and attaches to. This helps it activate PI3 kinase 4 signaling. P85 joins with P110 to form the P13K complex. P85 stabilizes P110 and regulates its activity, ensuring it gets activated only when needed, like when IRS brings them together during insulin signaling. This completes the PI3 kinase and activates it. PI3 kinase acts on PIP2 and adds another phosphate to it from an ATP molecule, leaving behind an ADP molecule. The additional phosphate causes PIP2 to become PIP3, which activates PDK1 and other kinases, such as PDK2. Inactive AKT, also called PKB, is a kinase that floats in the cytoplasm. It remains inactive until it binds to PIP3, where it gets phosphorylated. The recruitment positions it to interact and to be phosphorylated by PDK1 at the threonine 308 site. This partially activates the AKT kinase. AKT gets activated at serine 473 when PDK phosphorylates it. This, along with the phosphorylation at threonine 308 by PDK1, fully activates the AKT for signaling. This is the GLUT4 vesicle. This vesicle transports GLUT4 proteins to the cell membrane, where they form channels and allow the glucose to enter the cell from the bloodstream. Its movement to the plasma membrane is inhibited by the AS160 protein. The AS160 protein acts as a brake by keeping the vesicles from moving until it is phosphorylated and inactivated by AKT. AKT inhibits AS160 by phosphorylating it at specific sites such as 309, 642, and serine 588. This phosphorylation inactivates AS160's ability to block GLUT4 vesicle movement, enabling vesicles containing GLUT4 to move to the main membrane, where it is inserted to be part of the facilitated diffusion of glucose into the cell. This is glucose. GLUT4 transports the glucose into the cell from the bloodstream, reducing blood sugar. Glycogen synthase kinase 3 is a protein enzyme that inhibits glycogen synthesis by adding phosphates to glycogen synthase. Glycogen synthesis is the process of building glycogen, a storage form of glucose. This body links glucose molecules together in a chain, storing them in the liver and muscles for use later when energy is needed. When GSK3 is inhibited, glycolysis is no longer inhibited, which means glycogen is produced. mTORC1, a protein complex, inhibits IRS proteins through negative feedback, which dampens the feedback loop controlling the insulin-glucose pathway. An example of a disruption to the system is chronic overactivation of mTORC1. often caused by excessive nutrient intake or insulin resistance. This overactivation increases the negative feedback on IRS proteins, impairing their ability to activate P13K. The result of this weakened insulin signaling leading to reduced glucose uptake by the cells, higher blood sugar levels, and potentially the development of type 2 diabetes over time. Insulin resistance occurs when the body cells no longer respond effectively to insulin. Normally, insulin binds to its receptor on cell surfaces, triggering a cascade of signals that allow glucose uptake and the regulation of metabolism. In insulin resistance, these signals might be impaired, forcing the pancreas to produce more insulin to compensate. Over time, this can lead to hyperinsulinemia and eventually type 2 diabetes when the pancreas can no longer keep up. Thank you, and that's everything about the insulin glucose pathway.